So welcome everybody. I'm David Malkin. I'm a pediatric oncologist here at SickKids and clinician scientist, and also one of the co-directors of ACCESS. And we're really delighted to welcome Dr. Marianne Mateos here today. This is in just the day before, I think most of you are aware of the AACR Pediatric Cancer Symposium, which starts tomorrow evening and runs through the weekend that Toronto's hosting. So Marianne and a number of other Australian colleagues have made their way across the big pond and a bunch of land in the middle to arrive in Toronto, spend the time here. And so we're delighted to have you here. Just briefly, I think some of you may be less familiar with ACCESS. It's a pan-Canadian pediatric cancer network, which is advancing childhood cancer experience, science, and survivorship. That's what the ACCES is standing for. It's a combination, a network of researchers, healthcare providers, clinicians, industry leaders, as well as, very importantly, people with lived experience, so patients as well as parents of children with or who have succumbed to cancer. The idea is to bring all these entities together to really start advancing new scientific discovery, novel therapies, novel diagnostic tools to improve the lives of children with cancer and their families. So ACCESS is quite a substantial organization, if you will. And over the last while, we've had a number of webinars, and we'll continue to do so, hosted through the year with various areas of expertise and speakers. So today, we're excited to have this is our first ACCESS Presents webinar. It now has an official title. This is on the application of precision oncology. Dr. Mateos is a pediatric neuro-oncologist and clinical academic at the Sydney Children's Hospital in Radwick and the Children's Cancer Institute in Australia. I've had the honor and privilege to have visited on 2 or 3 occasions, an amazing conglomerate, if you will, of institutions there. We collaborate very closely with the Australian group and have done so now for a number of years. Dr. Mateos completed her clinical oncology fellowship at Sydney Children's Hospital and a PhD through the University of New South Wales, and as well followed that with a postdoctoral fellowship in neuro-oncology in the UK. She's been back in Australia since about 2019 and has been really playing a lead role in the Zero Childhood Cancer program, which is Australia's national precision oncology program, and is sort of, if you will, a partner or a sibling of, I think maybe the older sibling of Profile, which is the precision oncology for young people program here in Canada. Her expertise goes well beyond neuro-oncology, including precision medicine and toxicity profiling, which is incredibly important, and has co-developed a number of clinical trials in high-risk cancers. I don't want to spend too much time in the introduction because she is the one who we are here to listen to, but just to note that she's received the Kids Cancer Project Cole Reynolds Clinical Fellowship, which is helping support her work in high-risk brain cancers using sequencing information to better develop personalized treatments, targeted therapies, and novel technologies for detection of early relapse. So without further ado, I'd like to welcome Dr. Mateos, and looking forward to your talk. Thank you.
Thank you, David, for that really lovely welcome. I consider Toronto almost a second home in that I've got some very close friends who live in Toronto, and I actually did do a 3-month stint in Vancouver, BC Children's. So for those over in Vancouver, hello. So as David mentioned, I'll be talking about some of the learnings and some of the projects that we have running through the Zero Childhood Cancer program, but also more broadly with my research team. And it's really lovely to be the first ACCESS Presents talker. This is where David has visited. So this is the Children's Cancer Institute, which is based at the University of New South Wales. And very excitingly, next year, we're going to actually have a comprehensive children's cancer centre called the Minderoo Comprehensive Cancer Centre. And that will actually house both Children's Cancer Institute, it will house our hospital, Sydney Children's Hospital, and the Kids Cancer Centre, and also the University of New South Wales. So we really hope that that's going to help bridge some of the translational gap that we have and help move us forward. Before I actually start the talk, I usually in Australia start with a brief acknowledgement of country. I know it will be slightly different in Canada, but I really just wanted to acknowledge the First Nations people and to pay my respects to elders past and present. In terms of my talk, I thought I would structure it in a way to look at the areas that I think are emerging opportunities, but also ones that potentially there could be some collaboration projects going forward. If we look at the germline, I think it's definitely an area that we can harness more effectively. What I mean by that, and I'll expand on it, some of the projects that we have running, but also you have an amazing germline project here in Canada and particularly here in Toronto. Can we use the germline really more to better develop treatments? The next area that I want to be looking at is, can we use cancer genomics to better understand some of the treatment response and evolution that may occur during treatment? And can we develop slightly more novel liquid biopsy monitoring techniques? I will also be talking about clinical trials interwoven in my talk, but also specifically I will mention two of the current clinical trials that we have under development. One that's a close partnership here with colleagues in Canada. And then lastly, but definitely not least, is supportive care. And I have many different hats within that realm. My PhD was in clinical and genetic determinants of risk of side effects in ALL treatment. But particularly today, I'd like to talk through some of the treatment decision-making rubrics and how we, when we're moving forward with the novel precision medicine programs that are both here in Australia, but also internationally, how are we actually measuring who takes up those treatments? What are the predictors and how can we perhaps enable them or understand better about how we as a community take up novel technology? So I'll start firstly with the germline cancer risk part of my work. And before I really launch into that, I wanted to acknowledge, as we all know, that pediatric cancer, while incredibly important, we're often dealing with rare diseases with small patient numbers. And that's why more than ever, collaborative opportunities are really important. We also acknowledge that a lot of the treatments, many of them won't be successful. There's a high morbidity, but for those where it is successful, there is presence of lifelong toxicities. And as I mentioned, if we're using the germline, can we actually better understand methods to prevent cancer? That would be one really important area that we could move the field forward. High-risk leukemia, very important. I won't be touching upon that so much today, but it's definitely not a solved problem. Brain cancers, as you'll hear, there are some projects that we have looking at different aspects of how can we improve treatment and understanding of disease biology in brain cancers. And the other two areas, as I mentioned, we'll be looking at sequencing and application of novel technology, as well as what are our current pipelines for clinical trial development. I'll start with a project that began in 2015, so during my PhD, which was a side project. And what I mean by that, it was developed really with my interest in the germline, but it wasn't part of my PhD. And what I set up was a New South Wales-wide population study. So New South Wales is very similar to Ontario as a province. There are 8 million people there, so I imagine that's smaller than Ontario. But essentially what we were hoping to achieve was to enroll children with Down syndrome to then understand who went on to develop the pre-leukemia, so transient myeloproliferative disorder, and who subsequently went on to develop leukemia, so with some novel monitoring techniques. And I'll talk you through a little bit what that looked like. So in 2014, we published in Nature Reviews Cancer, that's with my colleague Glenn Marshall, Bill Weiss, and our team. We published on a couple of different embryonal-based cancers, and one of them was looking at TMD, transient myeloproliferative disorder, or transient abnormal myelopoiesis, which is a pre-leukemia. And we understand that in embryogenesis, that the first hit is trisomy 21. So the presence of a third chromosome 21 leads to proliferative excess, which is the first hit in this cascade. Secondly, GATA1, the mutation that's on the X chromosome occurs, and those two factors lead to the pre-leukemia. And we know that there has to be something that drives development of subsequent leukemia, which occurs in 25% of children. And Irene Roberts and others have done some very eloquent work really identifying cohesin genes and other factors as the third hit that leads to development of the leukemia. So we set out in our study to try and understand, could we better monitor the development and the risk of subsequent leukemia in our group of patients. We had 88 patients, so the recruitment actually was quite slow because at the time there was introduction of the non-invasive prenatal testing. But within our cohort, we actually did have some interesting findings. There are 2 of our review papers that are also listed there on the right-hand side. So the first finding that we had, and if you look to the left panel, panel A, so the variant allele frequency VAF of the GATA1 mutation is listed on the y-axis, and then the clinical age is on the x-axis. So essentially those patients who had Down syndrome TMD, so the pre-leukemia, are in the circles, and you'll see that most of them had resolution of the GATA1 clone, so it was no longer detected, and that occurred in the first few months of life. But actually, we did have one patient where it was still detected at 4 months of life, and then it completely resolved after that. They did not go on to develop the leukemia. And that information is actually quite novel from what we can see in the literature at the moment. The understanding is that 3 months post the pre-leukemia, if you have the presence of GATA1 PCR, then that predicts development of the subsequent leukemia. Whereas we're showing that at a very sensitive but low level GATA1, that really potentially we need to be looking at a later window, so 4 months before we can say that there's no chance of subsequent leukemia. The right-hand panel looks at the one patient in our cohort who had both the pre-leukemia and who went on to develop the leukemia. And you'll see that they had quite a high peak of the GATA1 mutation, which was just under 50% in the early prenatal, in the early postnatal life. It went down to almost 0, but was never 0. And then they subsequently went on to develop the pre-leukemia. So really, this is showing us that we need to be looking at a slightly later time point before we can say that they will not go on to develop the subsequent leukemia. And this is from our review paper that was recently published, which to me highlights some of the future research directions in this field in terms of Down syndrome associated leukemia, but specifically the role of MRD testing. How can we combine that with clinical factors and also full blood count monitoring to better understand those children who will go on to develop the leukemia? And at what point should we be considering potential intervention prior to development of the leukemia? So that's definitely an unanswered question, but you can see with the patient numbers that we're looking at, we really need to be building large collaborations to be able to understand and better implement this clinically. Now I'd like to continue along the theme of germline, but actually talk to you a little bit about my work in high-risk brain cancers and how can we better harness or understand the germline in this field. As we know, there are some high-risk cancers, brain cancers, that are sadly incurable. And one of them is diffuse intrinsic pontine glioma, DIPG, also now referred to as diffuse midline glioma, DMG. And this is an international project that I co-lead with David Ziegler, really looking to understand the prevalence of germline mutations in this cancer. To date, there's very limited data on the prevalence of germline variants in this disease, and we know that novel therapies are urgently required. When I started this project in 2019, 2020, at that point, there were a couple of publications which showed that there was approximately 7 to 16% of childhood cancer cases had a detectable germline variant associated. And what we also know from the very eloquent work here of Uri Taburi and his team and others is that if you use the germline in the case of constitutional mismatch repair deficiency, you can actually utilize immune checkpoint inhibitors very effectively with good treatment responses. So we thought that perhaps if we understood the germline of DMG better, we may have insights into both disease biology, but potential novel treatment strategies. So this is the collaboration that I established. You'll see from the map there that it includes Australia over in the very bottom right. We had both the German group and the UK group, as well as CBTN, so Children's Brain Tumour Network, and then colleagues in Montreal. Very keen to extend the collaboration, and hopefully after you see the data, you might be interested in joining it too. So what we started with was the collected clinical data from the respective groups, family history if known, as well as genomic information. So that was the matched tumour and germline. And that was using what was available, whether that be whole genome or whole exome, which was in the majority of cases. And there were only a couple of cases where panel sequencing was used for the somatic tumor material. And in that case, it was where the whole genome sequencing had failed. And patients were either DIPG or those that had DMG. As mentioned, there were various profiling platforms used. However, where the raw data was available, we actually put that through the bioinformatic pipeline used by Xero to ensure consistency of data calls. Where the raw data was not available, we looked at the curated variants from the other cohorts and confirmed through our manual curation process that they would be pathogenic or likely pathogenic germline variants based on our own calling algorithm. And importantly, these were pathogenic or likely pathogenic variants in known cancer predisposition genes. And we used both the matched tumor and germline samples. These are the international participating centres. As you'll see, some very large consortia, but definitely room for additional collaboration. There were 252 patients included in the cohort with a median age of 8 and roughly 50-50 male and female, with the predominant being H3K27M altered patients, mostly in the pons and brainstem and some in the thalamus. Typically, there was a poor overall and event-free survival of 11 and 6.5 months. And out of the cohort, 19 out of 252 patients, or 7.5%, had pathogenic germline variants. If you look to see where those pathogenic germline variants sit, so all the patients are depicted as columns in this oncoplot, and in the far left-hand corner, you'll see the pathways. So more importantly is the HR or homologous recombination pathway, where you'll see an enrichment of those pathogenic germline variants. So you'll see a few in PALB2, 2 in BRCA2, and a couple of others there. There were also, interestingly, 2 patients who had 2 pathogenic germline variants each, one in BRCA2 and a FANC anemia pathway gene, and you'll see they're highlighted in the red. There were a couple also in other pathways, mismatch repair genes. There were 2 patients. They were both heterozygote variants. And I've also depicted their base excision repair mut YH, which were not considered pathogenic germline variants for this analysis, but I've just included them for completeness. In terms of were there any associations between germline variants and the presence of other features, there was no association with the type of H3K27 variant, 3.1 versus 3.3, no association with location nor sex of these patients. What we wanted to look at is, were there somatic second hits in these tumours? And interestingly, so this was work that I did with Sam, one of our bioinformatic colleagues, out of the whole cohort. So once again, in the left-hand column of the oncoplot, there's both the germline pathway, and then below that, they're the somatic pathways. And you'll see there's one patient highlighted in the red who had a somatic second hit, and that was the patient who had the 2 pathogenic germline variants, one in BRCA2 and the second in FANCD, who had a second somatic hit in FANCD2. And this is a pictorial representation of where these main variants sat. And this is looking at the 3 key pathways that were found in this analysis. So the first is the Fanconi anemia pathway. So if you look at the number of variants in the DNA damage repair pathways, there were 4 out of 15 that were in the Fanconi anemia pathway, 9 out of 15 that were in the homologous recombination or HR pathway. And those ones, as we've seen in adult tumours, potentially could be targeted through PARP inhibition. The other smaller contribution that we saw, as I mentioned, were heterozygote variants in the mismatch repair genes. 2 out of 15 were seen in our cohort. So whilst they were not biallelic loss, you could argue potentially there could be a better understanding of the immune environment here and whether immune checkpoint inhibitors may be useful at all. So from this, I wanted to understand the somatic profiling of these patients. And so we looked at those that had full somatic, so somatic sequencing as well as transcriptomics and mutational signature data. So we had 4 patients where we had the presence of pathogenic germline variants and where we had these full data. And the first observation here is that when I divided it into the two patients who each had one germline variant compared to the two patients who both had two pathogenic germline variants, visually it appears that there are more copy number changes. So these are Circos plots, and you'll see in the middle here, I know that those online can't see, but essentially the green in panel C is copy number gains and the red is copy number lost. So visually panel C and D look a little bit more colorful, perhaps some more obvious copy number changes. But the way that we looked at this numerically is I stratified for those that had somatic TP53 variants versus not, and looked to see if there was an independent effect of the germline variants on the somatic profile. So the first observation here is of the patients who had two pathogenic germline variants each. You'll see that the panel D had no somatic TP53 mutation, but in fact, the double hit, so the two germline variants, meant that this patient had a higher percentage of genome-wide loss of heterozygosity, or LOH. And that means potentially these tumours were more unstable, so more genomic instability. And that was independent of somatic TP53 status, so lack of TP53 status. And similarly, in the patient that had a somatic TP53 mutation, they also clustered towards the higher end of the genome-wide LOH, implying, but definitely not saying definitively, that the germline variants could be contributing to the somatic profile of these tumours and increasing genomic instability. But certainly more work is needed to confirm that. The second aspect of this work is we wanted to look at whether you could assess the impact of BRCA1 depletion, so a homologous recombination gene. Could you assess the impact of depletion on the sensitivity of diffuse midline glioma cells to PARP inhibitors? And there were two shRNA constructs, so DOX-inducible lentiviral constructs, where BRCA1 was knocked down. And you'll see in the Western blots at the top, you'll see that the first construct worked with loss of BRCA1, and so did the second, loss of BRCA1 on DOX induction. And that also led to a reduction in cell colony formation, both shown in the agar plates here when PARP inhibitors were used. So the BRCA1 knockdown, cell lines, and here addition of PARP inhibitors led to a significant reduction in cell colony formation. And that's also represented in the bar graph for both cell lines, but not seen in the non-targeting shRNA. So that suggests that BRCA1 knockdown sensitizes DMG cells to PARP inhibitors. And then lastly, we looked at the cohort to see if there were any particular clinical responses. And we found that there was one patient who was actually the patient who had the double hit germline, so BRCA2 and FANC-E, who had an extraordinary response to PARP inhibition. The PARP inhibitor was actually combined with an immune checkpoint inhibitor. And you'll see here at baseline, this is the patient's tumor here. This is a multiply relapsed patient with diffuse midline glioma in the thalamus. And then subsequently, upon starting this PARP inhibitor-based treatment, there was a significant reduction in the tumor size, and here radiological resolution. So with this piece of work, this is, to our knowledge, the largest germline analysis in DMG, showing a prevalence of 7.5% of deleterious germline variants enriched in the DNA damage repair pathways. There was a clinical response to combined PARP and immune checkpoint inhibitors in one patient with a germline BRCA2 and FANC-E variant. And there's some laboratory modeling to show sensitivity to PARP inhibitors in BRCA1 knockdown cell lines. This is where I think we need further information to move this forward. So what are the clinical implications of this? How can we better understand the role then of genetic testing? I think it's definitely too early to be saying we should be doing widespread standard of care germline testing in DIPG-DMG, but I think we need more information to understand both what's happening at the RNA level. Some of these patients, and certainly some of the data we have with that patient who had the extraordinary response, is actually had complete loss of expression of BRCA2 and FANC-E. So there are some additional events that are occurring, and then with a larger analysis, I think we'll be able to understand where to from here. So This is part of the work that is funded by my recent fellowship. Laboratory validation is definitely important. The extension of the cohort and further analysis, including RNA sequencing and mutational signatures, I think will also be really important. And we're hoping to meet with Uri and Cynthia Hawkins and welcome other collaborations, potentially to broaden this collaboration. Next, I'll be moving to the use of liquid biopsy monitoring in children with high-risk cancer. As we know, brain cancer is the leading cause of death from disease in children in most developed countries, including Australia. And that spinal fluid, CSF collection, is really quite crude. We can look for the detection of cells, but that is about it. And increasingly, we're looking to see if we can develop more sensitive tests to enable both early treatment, relapse, so understanding of relapse, but also monitoring of treatment response. So the aim of this work was to develop a workflow to identify circulating tumor cells in the CSF. And it was really born from some patient experiences where I'd had a patient with, for example, a standard risk medulloblastoma, group 3, who had unexpected relapse post initial radiation. Widespread relapse, and this was with a gross total resection. Cytology was completely negative. And this patient very sadly went on to have some temporary benefit from chemotherapy, but eventually passed away. So it really begs the question as to what are the more sensitive tests we could develop to better understand early treatment resistance. And this is a large collaboration that I am lucky to be leading. And also I'm very pleased to be showing you some of our early data. And this will be a poster presentation as well at the meeting this week. So the workflow that I established with colleagues at Children's Cancer Institute and the liquid biopsy program was really to try and process these samples quickly. CSF was collected fresh in normal tubes and processed within 2 hours and run through a pipeline that allowed us to perform single-cell RNA sequencing, capture, and imaging-based analysis. That was then followed by library preparation and sequencing and single-cell analysis. I'm sorry about this slide, but I think this might be a Mac PC issue. But essentially what I wanted to show you here is that we have 14 patients in the cohort and we're continuing to expand. In the very fuzzy writing, you'll have to believe me, but we had several patients with medulloblastoma, ATRT, pineoblastoma, and ependymoma. These are all de-identified, but there were 4 patients with sequential samples already analysed, and we have more patients with sequential samples coming through. Our first observation was that those children that had larger cells present in that image-based analysis that I mentioned actually mostly had what we thought were putative CTCs, circulating tumor cells. And I'll show you some of the data which would support that they are truly circulating tumor cells. So you'll see in the first diagram there that there were no large cells, nothing over 15 microns, and this patient did not have any CTCs identified in the spinal fluid, whereas this patient was sadly one of my patients with very rapidly progressive group 3 medulloblastoma with MYC amplification, and there were lots of large cells identified, and many of these were circulating tumor cells. So we were able to, by incorporating imaging into the workflow, show that there were larger cells which were suspicious of CTCs. But to definitively identify them, we matched the single-cell RNA sequencing data with the bulk tumor data, so both the whole genome sequencing data and the RNA-seq. So the first observation here to mention is that we had to basically divide up the cells based on CD45 negative and positive. So the CD45 positive cells here are the immune cells, and the CD45 negative here are the ones that we're interested in with the CTC population. And you'll see that also, and apologies to those online who can't see my pointer, but essentially you'll look at on the far right, there are 4 genes there that we assayed for, that we looked at in the RNA sequencing: MYC, GFAP, OLIG2, and NeuroD1 for this particular patient. There were many more that we looked at, but you could see that the top area where these were the CD45 negative cells are the areas that map to the key transcriptionally highly expressed genes that were seen in a bulk tumor. So this was the patient with the highly MYC amplified group 3 medulloblastoma. And secondly, we also saw significant tumor intratumoral heterogeneity. This is also the same patient, which you'll see this is the bulk RNA sequencing on the UMAT plot. And then you'll see that on the very right-hand side with the 4 panels, that's the various level of expression of those different genes within the CTC population. So variable expression of vimentin, for example, but quite uniform expression of MYC. And to show you how I think this may be useful, but this is once again not an unfinished story. This definitely needs more patience. It needs a larger collaboration. I think it needs a clinical trial as well, is that this is my patient. This is shared with permission. A young child aged 18 months. This is not the picture of the patient, but a young child aged 18 months diagnosed with an atypical teratoid rhabdoid tumor, who also had a germline SMARCB1 pathogenic variant with biallelic loss in the tumor. Very poor predicted survival. I treated this patient with multimodal treatment, surgery, chemotherapy, and radiation. And this patient's now receiving oral targeted therapy with tazemetostat and is alive 2 years later. And what I wanted to show you was some of the early CTC data that we have, which I think is quite exciting. So it's in that top group, the one area with the circle, they're the CTCs. And you'll see over time, so this is from initial diagnosis. So panel B, from the very left through to the right-hand side, it's progression, it's over time. And you'll see that in the purple, that's the line that I'd like you to focus on. And there's an increase in the CTCs, and this is percentage, now at 0%, and this is the absolute cell count. So based on this data, and I still have another 6 months of treatment for this patient, there is no imaging change. This patient's clinically very well, and you could say that based on this CTC analysis that this patient is in a complete remission with no evidence of disease. So this is exciting data. It's very fresh data. I'm hopeful that in future we may be able to use this in a more extensive collaboration to really understand how can we use this data in a more real-time basis. So where to from here is going to be expanding the cohort and really looking at how can we harness opportunities to detect early relapse or improve our treatment, and also look at disease evolution, treatment resistance, and analyze this. And I think it is important that it needs to be done within the context of a clinical trial. The next aspect we'll be looking at clinical trials themselves and some of the developing clinical trials that we have, and also a focus on OPTIMIZE, which is a collaboration here with our Canadian colleagues. You may have seen this paper. This was Loretta Lau being first author, but our broader national collaboration through Zero. I can see Jordan in the audience as well, who was a fellow recently and is in Sydney and now here in Toronto. So this was a large collaboration nationally, but we showed 2 important things: that patients in our national trial do benefit clinically from precision-guided therapy, or PGT. And that is shown there in the red, that the patients who received PGT, there was a significant reduction or an improvement in progression-free survival. And there was also an objective clinical benefit for those patients who received PGT versus what we call unguided therapy. I think the challenge is, however, is not all patients are going to be receiving the treatment. And I'll go into that in a little bit later. But first, I would like to just give you a little bit of a flavour for some of the clinical trials that are under development or just opening. So one of the challenges is what to use for a biomarker, and we don't have to always have biomarker-driven trials. We can collect data and then try and retrospectively, if there are clinical responses, actually understand what might be a relevant biomarker. And so this trial is one that I co-lead with David Ziegler. It's a world-first clinical trial looking at a novel formulation of 5-fluorouracil in refractory and recurrent pediatric CNS tumours, and it's sponsored by ANSTROG, and also with significant philanthropic input from the Kids with Cancer Foundation. So that's an example of a non-biomarker driven clinical trial. And then we have OPTIMIZE, which is very much a biomarker driven trial that will partner with our respective national precision medicine programs, Profile here in Canada, and Zero in Australia. So several collaborators, several of you are in the room, but this is a really exciting next step to try and bring this treatment, precision-guided treatment, to children that we treat. And there are many not on this slide as well who are helping support this trial, so that we are now open in Australia at several sites and hopefully will be open in Canada soon. So the first arm of this trial, so if you look at this schema here, we have children on the respective 0-2 and profile programs. They have their tumour profiled. The National MTB provides recommendations. And then this in the far left is the cohort that is currently open. So arm A, which is paxilosib plus chemotherapy. There are going to be more basket trials developed and some that may be dropped at various times, but this is the general schematic plan for this. So paxilosib, what is it? It's a PI3K/mTOR inhibitor that crosses a blood-brain barrier. There's already a recommended phase 2 dose and maximal tolerated dose established. And so we hypothesized that if you did a combination of paxilosib with chemotherapy, that we may be able to enhance both the activity of paxilosib and improve patient responses. This is divided into both an early phase 1 and phase 2 expansion cohort. And what's important here is that one of the challenges is trying to develop a backbone that's going to be relevant for different pediatric cancer types. So specifically here, we've got one for high-grade glioma patients, which is just the temozolomide with paxilosib, versus other non-high-grade glioma patients where we're using paxilosib plus irinotecan and temozolomide. So the first thing to note is that cohort A2 is where they're clearly molecularly selected. So the phase 1 will be not molecularly selected, and the phase 2 will be molecularly selected. And we've chosen to include a cohort where there may be some clinical rationale to use this drug based on high throughput drug screening, based on RNA sequencing, and or other relevant mutations that may not be the key ones identified, but could be relevant for treatment response. And it's going to be 28-day cycles with paxilosib delivered daily and 5 days of irinotecan and temozolomide. And lastly, I'd like to shift through to the supportive care aspect. And I mentioned that precision-guided therapy, there may be a benefit in using that for patients. But I think what we need to understand is how many children are actually receiving these precision-guided therapies. What are some of the barriers? What are some of the enablers? And that may help us to more effectively use our resources within these programs. And so here, I'd just like to highlight a body of work that I am leading together with Glenn Marshall. And it's divided into a couple of sections. The first is a retrospective review where we've gone back to look at our Zero Childhood Cancer program, to look at how many children received a recommendation for a molecularly targeted therapy, how many took it up, and what were some of the factors that we could see that may have influenced the uptake of that treatment. Secondly is a systematic review, which is complete, but just being written up at the moment. And then there are some other aspects, including clinician interviews and a goal to develop a discrete choice experiment to really rank those factors and see what's relevant. And here we'd love to have further collaboration, potentially here and in other similar healthcare settings. So I'll be concentrating on the retrospective review data that has really been led by ACCESS-ACCES Senapati, who was also recently a fellow here at Toronto Sick Kids, and prior to that in the UK and Sydney. So ACCESS, so these slides next to are courtesy of ACCESS, but essentially we had a large cohort of over 440 patients. And if you work through down the diagram here, you'll see that the important thing here is there were 265 reports, and each report, as you may have seen, will often include more than one treatment recommendation. So we looked at those patients where a decision was made to either take up or not take up the treatment recommendation. And then we looked at the factors within each of those to try and better understand what predicted uptake of these precision-guided treatments. The first aspect here is that cancer type was statistically significant. And I think the difference really is that there was higher uptake within the CNS and solid tumour populations and less, so the no uptake is the dark blue, less in the hematologic malignancies. And that makes sense because they have access to a variety of other treatments, including monoclonal antibodies, CAR T-cell therapy, and transplant. The second aspect here is that the type of treatment the patient was on at the time of report was actually quite important. So patients who were on targeted therapy actually were more likely to take up a precision-guided treatment than those that were on conventional therapy. And if you look here, another interesting insight is if you looked at whether the patient was either in a stable disease setting or progressive disease, we know that patients who had progressive disease, uniformly, that was an independent predictor for uptake across all types of cancer types. But if you look here at the stable disease, what was really interesting to our team is that the CNS tumor group, so the no uptake here, they had a greater uptake of precision-guided therapy in the presence of stable disease. And I think that speaks to two things. Firstly, several of those patients may not have any other treatment options, or they might be on a targeted, an oral therapy or targeted therapy, and the clinicians may be more able to switch to another targeted therapy. And also the goal in high-risk brain cancer is often stability of disease. So that was very interesting to us. The reasons for a negative decision were varied, but you'll see the largest factor there was that they were receiving other treatment. So I think what a lot of clinicians will do is they'll enroll the patient, perhaps at initial diagnosis or relapse, have an initial treatment that they proceed with, but use the precision-guided therapy as the next step if that treatment is not successful. And here there was also a low percentage, less than 10%, where the drug inability to access the drug was the reason for lack of uptake. And so this is what Akansha had initially hypothesized, and several of those factors have actually been found so far in our work to be important. So progressive disease is an independent predictor. Receiving other therapies also an independent predictor. Through the retrospective data, we cannot look at expectations of therapy, but we're certainly interested in exploring that in the semi-structured interviews. Similarly, with the clinician factors, several of those are going to be quite subjective, cannot be gleaned from retrospective case review notes, but certainly can be explored in our semi-structured interview data. And then the tier of recommendation was significant and in terms of predicting who would take up the therapy. So this is really kind of the overview of what I wanted to talk about. I hope I've been able to give you some idea of some of the work that's coming through our program and some of the potential areas for collaboration. And really just to take home a couple of points. So firstly, I think we can be looking to monitor more effectively patients who have pre-leukemia and leukemia risk, and that's not just in Down syndrome, it will be in other myeloproliferative disorders, other AML cancer predisposition disorders. So definitely an area for opportunity and potential cancer prevention. How can we better harness the germline to develop new treatments? So we're starting to look at that in diffuse midline glioma and really keen to understand that better with a larger exploratory analysis. I'm very hopeful that liquid biopsy analysis will be a helpful tool for better understanding trajectory of disease response, but also which patients potentially are in a complete remission and could stop treatment versus those that patients who may need a little bit more treatment to try and completely lead to a condition or a complete response or a state of minimal residual disease. And I think we've got some exciting new clinical trials coming through, which I'm hopeful with our collaboration here will be able to provide benefit for children with cancer. And I think we also are just at the tip of really understanding within our respective precision medicine programs, what are some areas that may help evolve these programs in terms of who takes up the treatment decision, how can we better understand which patients do respond, but also how do we more broadly use novel technologies in our own practice. There are many people I would like to thank in this, many collaborators and funding bodies, but the most important really is the patients and their families. The large list here, some people I have acknowledged on the way through, but more broadly, this is the DIPGM collaborative. I'd like to acknowledge my mentors who have also been involved in some of these projects, driving them with me, including the PrEP21 Down syndrome and the decision-making program, liquid biopsy and zero colleagues, as well as my optimized colleagues, many who are in this room and who I've acknowledged in the previous slide, and the liquid biopsy team as well with the respective funding bodies. Very lastly, I would just like to put a plug. I did want to put some more pictures of Sydney at the very beginning, but I didn't want to distract you all with thinking about the beautiful climate and the Sydney Harbour Bridge and the Opera House. But I'm taking this opportunity to show you now and to explain some reasons why you might want to come down under, whether it's for just general travel, or if you are in neuro-oncology and want to come to ISPNO 2026, there is a little QR code to scan if you want to be up to date with what's happening. But otherwise, the Harbour Bridge, beautiful Sydney Harbour, this is the Blue Mountains, a koala, the beaches, and the Opera House. So with that, I would very much like to thank you all for joining in person. It's lovely to see you all. I'm very happy to take questions. Thank you.
Thank you, Marian. That was terrific breadth of work that's going on. Really exciting to see. So we're open for some questions. People are thinking, and online if anything pops up. Sarah, looks like you're.
Oops. Hi, I'm going to ask a neuro-oncology question, not being a neuro-oncologist. Just curious, for your DIPG-DMG collaboration, what was the proportion of patients for which you had a tumor sample? Because I think this is really shifted for those patients that we used to just diagnose them on a scan and not go further, and more is being published on the biology of this disease. And so just curious.
Thank you. Really great question, Sarah. So all of these patients had a tumor sample, which is a shift, as you say, from previously not biopsying. The challenge is actually having access to all that somatic sequencing data that could then feed into our algorithm. So out of the 252, I only had detailed sequencing information for 200 to be able to understand like the second hit, for example. But I think there's definitely been a shift over the last 5 to 10 years where we are most of the time biopsying these patients up front. Yep.
Well, people are thinking. So, of course, I have to ask a germline question, but in the DIPG cohort, you were doing paired germline tumor. Do you have any information on parents or cascade testing determine whether these inherited? The reason I ask partly is because a number of the studies in different tumors comes up with a very similar sort of 7% of these DNA damage repair pathways, and it's not entirely clear how much of those are, they may be pathogenic variants, but whether they're pathogenic in terms of causal of the disease, or whether they're just passengers that we don't really understand.
Yes, thank you, David. That's really key to that work. We did ask about family history if that had been collected in the respective cohorts, but it wasn't collected to a degree that is robust enough. We would have within our local Australian data access to be able to understand that, but what we hope is that in a larger collaboration, we can actually understand more the disease biology and the drivers versus passengers, because I do agree that it's not clear here if it's actually leading to development of the cancer or if we're just incidentally picking it up. And that's where I think it's going to be really important to do the RNA sequencing to look at the second hits, like, loss of expression, which may suggest that there's some potentially something more to those variants. But we're at the beginning of really trying to understand etiology versus passenger with the cancer. Yeah.
And I won't ask the philosophy of doing germline testing on everybody, because I think a few people here know what I think about that, and we can talk over a beer later on. Other questions?
Thanks for that talk. I'm wondering, for the analysis of the uptake, the decision making, were you able to look at any social variables and how that might have affected uptake of precision care?
That's a really good question. We would have some data, but I haven't seen it. I don't think we'd be able to really assess it in a robust way, but I think it could definitely play a role. It could certainly play a role in terms of understanding or the parental ability to want to engage in something like that. But at the end of the day, I think it does come down to the clinician presenting the data. A lot of the uptake is going to be guided by quality of life, what the parents want, whether they want intravenous chemotherapy versus targeted oral therapy, or a combination. I think we don't have really the data to be able to answer that question at this point in time.
Your proportions actually towards the end are very similar to what we have in profile in terms of why people may not have gotten a therapy, whether so in a sense it's actually kind of I don't know if reassuring is not the right word, but that the data from two entirely different cohorts are somewhat similar.
Yeah.
In that pie diagram.
Yeah. And I think actually the Canadian and the UK system is very similar to Australia. And I think it would be great to use this opportunity potentially to even involve your group with our group in terms of next stages of this project. So for example, the discrete choice experiment would be something that could be delivered. It doesn't have to be in person. Those things it can be online. And we're just in the process of really using our retrospective data, our systematic review data, and the semi-structured interviews. And in fact, if you look at the data for the systematic review, there were only 17 studies that were included in that. 15 of them really had the decision-making factors as a byproduct of the paper. It was a secondary objective, and there were only 2 that really addressed the question. So I think there's a lot of potential to understand this area better.
Okay, there were quite a large cohort of people online, so thanks for those who are online. And I think if there's no other questions, we'll let you free for a couple of hours before dinner. And thank you everybody for attending, and looking forward to seeing people who are attending the ACR meeting tomorrow. Okay.
Perfect. Thank you very much, everyone here and online. Thank you.